SANDHAN, All Gujarat Integrated Classroom.
સેટેલાઇટના માધ્યમથી જોડતી કડી એટલે સંધાન.
Good morning, students. Today we are going to discuss about the methods of the gene sequencing. As we all know that language of the life is basically a language in which everything is coded in the form of the gene. And this gene is basically having all the information regarding the entire part. So first of all, we'll be just going through the basic that what is the gene. So basically, suppose we are starting the entire journey of the life, then it will come from all the observable characters that we can observe. And these characters can be coded by the different part in our body. And this coding is done by the gene. So that way, a gene is a basic physical and functional unit of the hereditary. And chemically, this is made up of the DNA and act as an instruction to make all the molecules which are called protein. And after that, these proteins are responsible for carrying out all the different kind of activities in our body. And that way, all the characters which we are observing in a phenotypic way, all the phenotypic characters are ultimately coded by the gene. Thus, if we can have the information about the DNA sequence of that, then we can have the lot of the information regarding the gene. And today now, besides the gene, the entire gene sequencing, it has gone up to the level of the genome. And now it is possible to have the complete gene sequence, and even we can have the complete genome sequence. And this is having the wide application in the different areas in the field of the microbiology, biotechnology, and all areas of the life sciences. For example, we talk about the DNA sequencing. Then what this DNA sequencing is? Basically, the term DNA sequencing, it refers to method for determining the order of the nucleotide bases. As we all know that it is basically made up of the 4 nucleotide: adenine, guanine, cytosine and thymine, and that is creating the entire molecules of the DNA. Always the A and T that will be creating the bond, and even the C and G will be also creating the bond. And orientation of this ATGC on the entire sequence of this DNA, that is basically responsible for all the functional aspect of the gene, whatever we have discussed earlier. So that way, the process of the DNA sequencing, it will determine the entire sequence in the form of the A, G, C and T, and that is given to us in the form of the linear sequence that can be analyzed by the life scientists for the different application of our own interest. Suppose we are going in the historical development of the entire method of the gene sequencing, then we have to go very long back. Even though the technique was started in the 1970, at that time the techniques are not that much good as we are having the today. And at that time people have gone for the gene sequencing with the simple method based upon the 2D chromatography, and that was in the 1970 that some of the laboratories were working in the field of the DNA sequencing, and it is taking very long time and even very short output for the entire method. So in the 1970 the first DNA sequencing was obtained. After that, in the 1953, the Watson and Crick, they have discovered the double helical structure of the DNA, and that has changed the entire world of the DNA. Now we are knowing entire details of the ATGC, how it is getting bound with each other, and whatever the 3D structure of that was known to us. Similarly, after that, from the 1977, the Maxim Gilbert and the Sanger independently they have developed the methods for the sequencing of the DNA, and that method has ruled up to the 10 to 20 years in the entire DNA sequencing science. And both of this, it is having their own different kind of the mode. But even till today, we are also using the method which is developed by the Sanger. So in the next part, we'll be just taking the details of the Maxim Gilbert and even the Sanger separately. In the 1975, with all the techniques available for the DNA sequencing, from that only the first DNA genome sequence of the bacteriophage X174 was done. And that was the first organism for which such kind of the entire genome sequence was available. So from the 1975, slowly the concept is came to the entire genome sequencing. In the 1985, the revolutionary development is done in the field of the molecular biology. The Curry Mooley, he has discovered the PCR, and that was the technique for the in vitro multiplication of the DNA. So whatever the living systems are there, that is creating the multiple copies of the DNA. Similar to that only, with the help of the PCR, in the in vitro condition, in the lab, we can create the millions of the copy of the DNA. And that has changed the entire world, not only for the molecular biology and even for the sequencing. Based upon this only, in the 1986, Hood and Smith, they have developed the first automated DNA sequencing machine. Because the DNA sequencing machine, it was having a very troublesome and long protocol, and without automation, whatever the output of this DNA sequencing machine was there, that was coming less. So only after 1986, the automation was entered in the field of the DNA sequencing. In 1990, the Great Human Genome Project was initiated, and along with that only in the 1990, development of the pyrosequencing was also done. But even though at that time the people were using the method for the DNA sequencing, that was the Sanger only. In the 1995, the Venter, they first published the sequence of the Haemophilus influenza, and that was having the size of the 1.8 MB. So after viruses in the field of the bacteria, that was the genome that is first came. In the 1996, NIH funded 6 group attempt to the large sequencing of the human genome. So that way now people have entered not only from the bacteria, but they enter up to the level of the human to get the entire decoding of the DNA sequencing. In 1997, introduction of the mega-based capillary sequencing machine is came, because whatever the earlier machines were there, that was normally using the electrophoresis technique, and that was taking a quite long time to analyze the data. And from the 1997, when the capillary sequencing was invented, the entire process of the sequencing that has become more rapid and even more accurate. In the 2001, by the effort of all of this, the Human Genome Project Consortium published the first draft of the human genome. So that way, the entire human genome sequence was available to the world in the era of the 2001. 2000 onward, the entire sequencing technology has changed revolutionary. Whatever the sequencing technology was used till now, that was known as the first generation or the basic sequencing technology. 2000 onward, the 454 sequencing or the second generation sequencing has came in the picture, and with the help of this, the sequencing speed has gone increase, and a lot of bacteria and viruses are getting screened by this entire system. In the 2005, the first 454 next generation sequencing system GS20 was commercially available for the market. In the 2007, first applied biosystem, they have developed the next generation sequencer with a different kind of the chemistry, and that was known as the solid. So that has also changed the entire phase of the competition in the next generation sequencing. In the case of this 2005, the Sanger was the only option, but after that, due to that only, the price reduction is came, and now till today, lot of new platforms are available, and due to that, even the cost is also getting reduced along with the increase of the speed of the DNA sequencing. In 2006, Illumina-based first Solex system is came, and from the 2011, the first iron torrent system is came, and from the 2009 onwards, now we enter in the third phase of the DNA sequencer, and in this only we had the first helicose single molecule sequencer. Similar to that, in the 2011, the first PEG bio single molecule sequencer has came in the picture, and even till now the Oxford has also produced the nanopore technology, and that is also under rapid development, and soon that will be also available in the market for the rapid sequencing, and this all are now in the era of the third generation next generation sequencing of that. So that is the short history of the entire sequencing machine. Now suppose we are starting with our earlier part, that how the sequencing was done. Then even though today we are having a lot of advanced technique, but to get the advanced technique only, we have to go through the entire basic technique one by one. So we'll be starting with the very first technique, which was historically developed by the Max and Gilbert in the 1977 in the Harvard University, and that is basically depending upon the chemical methods only. First of all, whatever the sequence of the DNA you want to sequence, that sequence would be radiolabeled with the end product, and this radiolabeled nucleotide would be added into that, and due to that only, now whatever the fragment of the DNA would be there, that would be having the radioactivity, and that can be detected on the electrophoresis very easily with the help of the autoradiobiography. So that was the first step, DNA polymerase neo fragment that is incubated along with the CTP, DTTP, DGTP, and even the radiolabeled DATP. After that, whatever the sequence of the DNA is there, that fragment would be chemically treated, and this will create the different kind of the breaks, and this small proportion that will be creating the 4 different kind of the breakage, and that 4 nucleotide base in the each of the 4 reaction would be created. So basically the entire DNA that would be getting broken specifically at the 4 different places. You can have the break at all the places wherever the G is there if you are using a specific reaction mechanism. If you are using a second kind of the reaction, then you can have the breakage wherever you can have the A or G. Similarly, you can also have the breakage at the C, and you can also have the breakage at the C plus T. So the entire chemical which is used like this, suppose I am treating the entire DNA with the dimethyl sulfate, then dimethyl sulfate that will be going for the base modification, and after that with the help of the piperidine, that misplaced base would be displayed, and from that only the entire strain would be getting separated, and both the fragment of that is also getting seized, or that is getting separated with the help of the piperidine treatment. So this is known as the chemical cleavage of the DNA molecules. But the advantage of this cleavage is that the cleavage is highly specific in nature, and at a specific nucleotide only you can have the breakage. Now, depending upon the different reactions or depending upon the different kind of the chemicals, you can also control the cleavage. For example, I want to cleave the DNA wherever the G is there, then I can use the dimethyl sulfate for the chemical for base alteration, and after that base alteration, whatever the alterated base is there, that is removed with the help of the piperidine, and again for the chemical strain cleavage also we can use the piperidine. Whatever the figure earlier we have seen, that was mimicking the same thing. Similar to that, suppose I want to cleave the DNA wherever I am having the A plus G, then we can just cleave it wherever it is having the chemical alteration, and for that chemical alteration normally the acid treatment is given. Again for the alterated base removal, the second acid treatment is given, and finally for the strain cleavage piperidine is utilized. Similar to that, for the C plus T, hydrazine is utilized, and after that for the alteration base removal we can utilize the piperidine, and again for the chemical strain cleavage similarly piperidine is utilized. Similar to that, suppose I want to cleave specifically at the C, then for that hydrazine and alkali is utilized, and again after the following the treatment of that base alteration with the help of the piperidine, you can remove the alterated base by that mechanism, and this chemical will be utilized piperidine similarly for the strain cleavage. So with this entire thing, suppose we are observing the entire protocol, then that would be going like this. Whatever the duplex DNA is there, then first of all that would be going for the radio label. After radio labeling, that would be cut with the specific kind of the RE system, and after that that would be denatured only with the end label strain, and that would be visualized on the autoradiography. So you can see that with the treatment of that we can have the fragment which is either ended of the G, or it is ended with the A plus G, or it is also ended with the C plus T, or it is ended with the C. Now whatever these fragments are generated, that fragments would be going for the entire process in the next part as it is radio label. Then after that with the help of the entire electrophoresis we can go for the size separation of that. So when we are running the gel electrophoresis, due to the presence of the radioactivity, all the fragments will be detected in the autoradiography, and that will be going to be separated according to the size. And this gel is exposed to the X-ray film for the autobiography, and that will yield a series of dark bands, and each corresponding to the specific radiolabeled DNA fragment from which the entire sequence is going to be inferred. So the entire process would be going like this, that suppose I am running the entire electrophoresis from the tube 1, then that from the tube 1 whatever the fragment I will be getting, that fragment would be having the end nucleotide G, and depending upon their size that is getting separated. Suppose I am running the tube 2, then that will be creating the bands for the A and G, for the tube 3 that is for the T and C, and for the tube 4 that is C. And after that when it is going to be arranged together, from that arrangement only we can just find out that what is the sequence that is G, T, A or C. So that is arranged in a similar way, and from that only the entire sequence is getting inferred. So that was the technique that was utilized in the first phase. The basic disadvantage or the limitation of this technique was that basically it is technically quite complex, as it requires the 4 different kind of the reactions, and even it requires the similar treatment of the 3 different cycles. Lot of hazardous chemicals like the acid, base, piperidine is utilized, and due to that handling of that was quite difficult in the lab. Again it was quite difficult with the scallop. It can be utilized only up to the small fragment, and that cannot be customized in a standard molecular biology kit. So that was the reason that this technique was not become that much popular, and even that is not that much used in the field of the molecular biology. And due to this only in the earlier part, even the sequencing technology was in a premature stage, and even we get the lot of few sequence with this entire technique. Again, the second kind of the technique is developed parallel to that, and that was developed by the Sanger. and for that Sanger also got the Nobel Prize. The Sanger sequencing, it is utilizing the some other kind of the chemical technology for that. It will be utilizing the di-deoxy sequencing or the chain termination technique. And the key principle was the use of the specific di-deoxynucleotide triphosphate as the DNA chain terminator. So along with the development of the PCR, now it was possible to create a multiple copies inside the PCR. So what these people have done is, they have just gone for the PCR reaction, and along with the PCR reaction, when we are adding all the DNTPs, they have also added the di-deoxy DNTPs. According to that only, whenever this is going to be incorporated in the DNA, that will terminate the chain. And from that chain termination only, you can have a similar product as we had in the earlier chemical cleavage, that we can have a different length fragment, and that is specifically ended with a specific nucleotide, and that can be analyzed further. So that was the basic principle behind the entire chain termination method. The basic requirement for the sequencing is, as we have seen earlier, that basically it is utilizing the PCR technique. So you require a template DNA. Along with that, as we want to run the PCR, we have to use the label oligonucleotide primers. And again, you should use the heat stable DNA polymerase. And that will create the entire polymerase reaction in the in vitro condition. Along with that, we are using the ddNTP and dNTPs. So you can see that basically both of this, that is having the only difference of having one oxygen less, and that is creating the entire chain termination reaction. So when we are just going for the entire reaction, along with all the basic things, we have to also supplement the required PCR buffer, and we have to go for the repetitive cycle as per the need of the entire cycle. So normally it is going for the running around 30 cycles for that, and like that only the entire PCR would be taken for that. What will happen when we are going for the entire PCR reaction? So first of all, a single strand of DNA to be sequenced, that is shown yellow color, that is hybridized with the 5' labeled synthetic di-deoxy primer, which is shown brown in color. So now you can see that the primer is going to be bind with that. It is radiolabeled, as it is having the some kind of the fluorescent that can be also detected. In the next step, the primer is elongated with the use of the DNA polymerase in the 4 separate reaction mixture. Each tube will be containing the 4 DNTPs plus 1 di-deoxy triphosphate, and the concentration would be 100:1. So according to that, when we are running the reaction, we can have the one tube in which specifically it will be just getting terminated as per the addition of the DNPT. Suppose I have added the DDGTPs, then the reaction would be getting terminated at the G, and similar to that only, I can have the specific cleavage wherever I am having the A, G, C. So similar to that, entire reaction is done. So just we can see the same thing. DNTPs are added along with the DNA polymerase in the first tube.